Yeah, so you can keep it at aside. Let's have some basic talks. Yeah, for the microbiologists, if you are using the press code or the pulsar, that is very worth for the bachelor students. Pulsar book, maybe you are going to purchase maybe about 400, 500 rupees only, not more than that. If I'm not wrong, it is very thick. That can be useful for the up to masters also. From the market, you can purchase. Some PDFs are available. If you're going to take out their prints also, it may cost you around 800 rupees because the 1000 page book is there. To purchase the 500 rupees, it's worth because the color printing is also there. So you can keep that book with you. Microbiology by Pulsar is really worth for the bachelor students and maximum points for you people, not only this paper. So many others also related immunological aspects are coming for the cultures, some IPR related parts, and so many things are going to included over this. Even for those who are looking to further do like a DMLD, those who are looking to do pursue some of the parts. So pathogenicity related some special chapters are there for the several diseases are there. So that paper are also included over it. It's a full zone, one kind of a book, and easy to learn for those who are initiator. Sometimes may happen that as a microbiologist you have started the right now, right? Just first and second year you are to understand and learn the basic concepts is required to be in a brief part should be started. So other books are there. It may be more in a detail sometimes, and it make a much cumbersome for you people to start up. Once you go through the pulsar, you may move further ahead for the other books. So I will recommend you people that you may purchase that pulsar. Okay. It's maybe not more than that. If you are going to take it out, certain print out 200, 300 rupees, you are just investing, right? You are not going to purchase original book. Okay, but it is the Indian edition. Indian from the Asian countries, especially, that is a very on a low price editions it is. So you can purchase it in maximum hour. This paper, other than this paper, about your 50 to 60% syllabus will be there from it. Okay, 50 to 60% syllabus generally it's get covered. Same like it's enzymology, it's there. Maybe they have initially given in a brief. So same part you can understand and learn in a brief level, then you can move ahead for a detail. So it may be like that those who are learning by themselves. For the self-learning, it is far better. Or you want to just to complete their gist or the essence of that area, then you can cover up it very fast level. Sometimes you are looking to learn many things, but it is required more time. For the same unit, you can cover up in the 3 to 4 page. For a detail, you may go for the other pages or the other books is better. So just going to say that Pulsar, you may purchase about it. Regarding our just today's topic, that's related to methods for the aerobes and anaerobes. So basic some concept related to aerobes I have talked. In this point, we are going to talk more in detail about the anaerobic things. Okay. So first will the aerobes related. So here it is what? That a bacterial culture, either of the two way we are going to get. Either it is in a solid or in a liquid broth. So liquid, it is like you have kept your tube, you have performed your experimentation in a tube. So what we have did, the tubes were kept into shaker. The shaking conditions will provide more aeration because from the cotton plug, the air can pass. It will be a filter. It not allow other bacteria to or microbes to get past, but air will going to get past very easy. So aerations will be there. So chances of the bacteria, those who are in aerobic conditions able to grow, will more get facilitated. Other than that, if you're working in the fermenters like a bigger jars. Okay, it may be at a lab scale or the pilot scale or the industrial scale. When you're working, there is a called the system called the agitator and the aerator. That fermentation technology bioprocessing will be there, one of the unit. What generally done over there? It is like just think like a washing machine. We know that at the washing machine, at the bottom, something is continuously rotating. Right? To rotate all the clothes, something is rotated. The same like that, it's called the agitator. In the fermenter, same it is the agitator. It's like a washing machine is going on. Everything is on keep on rotation. Other than that, within that, some tubes have been kept. It's like aquarium. We know in aquarium, for the aeration, to increase the oxygen level in a water, what is been kept? Some tubes are being kept. So continuously bubbles will start. coming out of it. So that processing is done in the fermentation technology. Some tubings are being kept within inside. So continuously it will start to give the bubbles. The bubbling method is going to provide the air within our system to our bacteria and continuous rotation, because you cannot rotate all the big jar from the outer side. So what we can do within that side, we can rotate. So there is a RPM that is also fixed. You cannot go beyond some limitations. Otherwise, the bacteria within the medium get collapsed and get destroyed. So that rotation of that agitator, it's not like as much as fast as a washing machine. There we are looking to fight. Okay, that is a fighting is like a dust particle gets should remove. The speed is very high, larger, but here the speed will be at their optimum condition. So agitations and aerations are two important phenomena are there. It's been done for the aeration. So what we are going to look or understand over there, we want to provide the air to our bacteria when it is aerobic condition. Okay, so this is the basic concept. It's been done that in atmospheric conditions, aerobes can grow very easily. If air is not there, no oxygen availability, then this kind of bacteria are going to die very soon. Okay, it is like as we are the aerobic conditions liver. Okay, if the oxygen is not there, without oxygen we may going to die. Same it is there for the microbes. Without air they won't survive. Okay, yeah, some are the facultative organism. We are not just currently looking for it, but facultative organism means they switch on. Without oxygens they can move into anaerobic condition, and then they can sustain their life. So aerobic and anaerobic, two possibilities are there. So currently we are just going to talk about the aerobic and the anaerobic. Strict aerobes, strict anaerobe. Such terminologies are coming. Okay, the strict anaerobes will going to die without oxygen. Strict anaerobes will going to die in the presence of oxygen. So some methanogens are there. If air is coming, they are very much sensitive. In the presence of oxygen, they are going to die very soon. So it's depend. So this can be achieved either by vigorously shaking the flask or tube on a shaker. Okay, one important general. In a flask, we can prepare the culture, and that flask is being kept into the shaker. Okay, I think you have seen the shaker, right? Last also, I have kept some tubes over there in during the lectures. So shaker is like there at the bottom. Their base is continuously rotating, and over the rotations, what we have kept? We have kept their stands where we can put our flask. Tubes can be kept, flask, jar, beaker, whatever your experimental part there, we can firmly bind it. And there also, we may use the rubber band. Sometimes may happen that while keeping the flask, there are the on a base, on a shaker, different kind of adapter has been given. It is like a flask is there. You have started with the 250 ml. Sometimes you may work in the 100 ml. Sometimes you are looking to work in the half or the 1 liter. When you went to the shaker and you are trying to keep the 1 liter fast, you will realize that, okay, where to shift, because there is no such direct facilities are there. It is like you are working at your home at refrigerators. Okay, and within the refrigerators, you have kept their shelf at one particular place, right? And sometimes some guest has came and you have purchased the milk about a 5 liters, so the 10 liter is there. You want to keep that larger bowl within there. What you need to do, you need to remove the certain shelf to keep your larger bowl within inside. So these are the mechanisms are that how to handle the shakers. So you need to change their adapters. So for that you need to use some screw. Okay, screwdriver is there to remove the previous that adapters to keep the only 250 ml flask. Now you are looking to keep for the 1 liter. So within there also several kind of adapters are coming. So that is a technical part. While working, you are going to come to know about it. Okay, and the second point is what bubbling sterilized air into the medium. Yeah, most important sterilized air. That is passed through filters. So there is no other microbes are there. Sterilized air is required to get fed. Otherwise, it get contaminated. Contaminate our total jars. So our main principle is what? To provide the oxygen. That's the main our agenda in case of our aerobic conditions. So atmospheric air is there. when you are working into the open space. When we can work into open space, that is on experimental level. When you are reached to the higher level microbes you have already isolated, you want to decontaminate several sites, specifically into sewage treatment and others. So over there, you cannot have the total closed system. So your microbes should be able to sustain and manage themselves at the lower temperature, higher temperature, or they should be able to manage their aerations within the lower or the higher level. Okay, so there we need to pump the atmospheric pressure even. With atmospheric air is fed, but that only able to manage when your microbes are potential to do your work. So when you are starting your work, we need to give the sterilized air only for our arounds. And the sometimes, yeah, when you are working with the solid plates, liquid is okay, solid plate is there. What it is required, I may provide some of the times specific kind of a parafilm. Okay, parafilm. Maybe you have seen that there are so many beautifications. Especially, parafilm is used to remove their hair. That is the one creams or parts are coming that has been first stuck over there. After they solidify, you may rub out it. Okay, that is some materials are coming. So parafilm is like a material. Okay, which is get liquidified, then they get solidified over the surface, or you can stretch it also. Okay, so such part is being used in case of a particular beautification. So they are coming in a strip. Such strips are being used to close your lid. That is like a Petri plates I do have. Okay, when I have kept such Petri plates at the certain place and I want to store that plates at one particular area, so it may happen that by chance it get opened. So what I need to do, I want to keep them at the particular place safe in a conditions. So generally what we are doing, at they are can be used like a candle. That is, the candle is being used. So drop by drop, if you are going to put their candle drops on their lid, so it's going to get solidified over the surface and you can save it. It is like at a home, sometimes you are preparing some ketchups or some particular things are being prepared. You want to lock it. So what you are going to do, you may continuously move with the cello tabs, but cello tabs won't going to stick over there. So what are the another options are available? You may use some liquidifying materials. Sometimes you are knowing that at a home, plastic bucket is totally get collapsed, or the something is happened, or crack is there. So some people are coming from the outer side to make it for having the pest-like material. Or sometimes you are going into some grocery shops. They are giving the some tube-like or the pipe-like is given. When you are making them hot, it is like a plastic, or you can say the ball pen is there. Sometimes during childhood, maybe you have burned your ball pens. What's going to happen? While the burning the plastic material, it's going to put it down with the drop by drops. Right? That part you may have done. But drop by drop is done, this polymer is different. So some special polymers are coming. When you melt it, and drop by drop, when you're going to post over any of the area, it is going to get solidified as per your requirement. So drop by drop, you can fall it down. When you're looking for the aerobic and anaerobes, or you want to preserve them, so you want to close their lid. So closing of the lid required to firmly close with using certain polymers. So some polymers are being used into it. The paramil is a kind of a natural polymers. They are coming in a strip. So you can drag it. Elasticity is very high, but not that much. Okay, so I will show you later. That is how to use the paraffin. So with using the paraffin, you can seal it. Okay, but when you are looking for the aerobic conditions to be get maintained, we are not going to firmly bind with the paraffin. Otherwise, what's going to happen? The bacteria cannot survive. So that closing of their lid is generally done during the aerobic conditions to move in the anaerobic condition. Right, so such part are there. So how we can acquire these aerobic cultures? So one is the very well-known liquid culture, keeping the things in a shaking condition. Okay, other than this, other three plates method we have already learned: stick plate, spread plate, and pour plate methods. Right, each maybe you are knowing. Sticking, you open the plates, you are going to stick with your wire. Okay, pour plates. In a pour plate, you are going to entrap your microbes within the agar. Then you are going to pour. And the spread plates, you are going to use the glass rod to spread your microbes on a plate. So it is related to solidifying media. Okay, to get the isolated pure culture into it. Agar slant. Here you can see within the tube. Here it is. It is agar slant. So slant means what? When you are looking to solidify your medium, if you are going to keep the things on a vertical conditions, when you have added your agar within the medium and you are going to keep in a vertical conditions, it is going to get solidified within that. But you can going to slant at a particular 45 angle and you keep for a half an hour. What's going to happen? The solidifications will be there, but into at a one particular angle. So surface area will increase. Our purpose and motto within the tube, availability of the area for the streaking part is going to increase. So then after we are going to streak over it. You are going to may see these lines are there. So streaking on a plate is done. Similarly, streaking is done on a tube. So even a sugar tube, you can perform in a test tube also you can do. But it requires the practice. Otherwise, what's going to happen? Just you may insert it, you may poke into the agar itself. So one has to do a certain level of practice to insert your loop and you may going to streak over this. Okay, so this is also basic called the agar slant method for the aerob to grow. Then the dispensing the medium in a shallow layers, providing the aerations by constantly taking the inoculated liquid culture. That is mean in a shallow cultures. Okay, in a liquid cultures, you may perform the same. Sometimes it is done in open also. It depends upon the bacterial culture or the mixed culture is there, and we are looking to grow only aerobes. You can do same experimentation in open house also. But open house working is done. Sometimes it may not give the as result what we decide, because there is a chances of a mixing of the conditions possibility. Okay, but it is performed when we are working into the sludge treatment or the wastewater treatments. In such cases, there the material itself it is contaminated, raw material it is contaminated. So over there we may go for such things, and over there also we may use the sparger. Sparger, it is nothing like the pipe like structure. Hollow pipe is coming and from there air is coming out. Okay, so it is like a aquarium. Within the aquarium, you know that the pipes are getting open. Okay, so many kind of a beautiful spargers are available. You may go in the aquarium, they may provide the different kind of things. Right, same is people are using in the fermenter or in agitators or in the sparger. So types of spargers, what are they? That you are going to learn in the bioprocess technology. Some papers are there into your syllabus or related to fermentation technology. Maybe in the next year, in third year it is there. Okay, the part 2 is the anaerobes. So till now, anybody having any queries related to aerobes? How to grow aerobes and what are the basic need of aerobic conditions? So basic need is what? Provide the oxygen. How we can provide the oxygens? Either you can keep the things in the staking conditions or you may provide the air by bubbling inside. Dispensing the medium in a shallow layers. So continuously, what going to happen? That is been transferred. Dispense the medium into shallow layers. It is like the here the surface is there, one container. So you may transfer here again to the second. It is done the dispensing from one to another. It is like a soda bottle is there. Okay, what he is generally doing, it is mixing into one to another. So within shortest of the time, that is going to bind with the outer side air. Why? Thumbs up. When it is in closed system, not going to happen. So the bubbling is not started. This whole carbon dioxide within there, it is not going to react. Right? Okay, when it is coming in the low atmospheric pressure, then only. So when you are going to shake it well for a 15 to 100 times, but till you haven't opened your bottle, it can remain as such. Okay, so it is the same thing, dispensing in a layer by layer. So it is, you may transfer from one place to another to another. It's only possible when you are not looking for a sterile condition. Then it is possible. You want to make aeration. You want to grow your bacteria. So you just for the aeration purpose, you may transfer one glass to another. Okay, so we are looking to provide the air to our bacteria. So dispensing in a cello layers. Generally, it is in a cello layers. You can do in a glass also. It's depend. Even you can use the flask. That is a shaking is done. Same it is there. You are going to shake from one to another. Okay. Any questions other than that? So it is aerobic conditions. It is nothing but to provide the air. Now the second targeted part, it is anaerobic bacteria. So it is related to as much as without oxygen, but it won't going to happen always 100% without oxygen to get. It is really difficult. So we may go for the some basic options availability. Either you may use a certain media in a way that it having the as much as less oxygen components are coming. Otherwise, you may use some other options or technology to reduce down the oxygen. So what are the basic things could be possible? Like when you are boil your water on a gas stove. After a certain while, you component some bubbling is started, right? Bubble is started to come out of the water. So from where the bubbles are coming? Can anyone say? Bubble is start to come, right? So air was there within inside? Air was there? Huh? Okay, so from H2O, the oxygen is start to release. You are going to say. Do you know about the other? Yeah. Yeah. Very good. Okay. The question is that when you are going to keep your bowl on a gas stove, just fill with the water, and you are keeping for the boiling. After a few minutes, it start to have the starting of the bubbles. Generation of a bubbles is there. Why? Okay, so it is a bubbling is going to form. That is by the layering it's move water. No. Movement of the water is there. Okay, hot and cold. It's yeah. So dissolve just not only of oxygen, dissolve gases like a methane is fully get dissolved within it. Right. If the temperature may rise, like we are now knowing about the global warming, right? Methane is also get dissolved within the water. If the temperature of the sea level may rise, then the methane may start to come upper side and it may start to have the direct fire on their surface. Okay, then the temperature may going to rise. It is like a theories are there related to global warming, right? The same it is there. It is having the highest capacity like a carbon dioxide, oxygen, many gases are get dissolved, remain dissolved in a water. Maybe you are knowing in environmental microbiology or some of the papers are there. You are working in the COD or the BOD. Maybe you have learned about that, right? So oxygen demand that maybe you have learned. So oxygen is remaining dissolved form. So we need to reduce it. So first, what we can do, boil your media for a continuous some time period. So what's going to happen? That the bubbling is going to cause all the oxygen, dissolved oxygen, and other gases get coming out of it. That's the main, a basic criteria we need to perform when you are looking for the aerobics to anaerobic bacteria to be get cultivated. So first is the media preparation. Okay, media should be there in a such a way to be get prepared that as much as less amount of oxygen is there. You cannot make it zero. Okay, you cannot make it zero because at the certain time period, once again, from the air, oxygen may start to get dissolved within it. But yes, you can do such thing in the certain in the closed conditions or the reduced conditions or that a low atmospheric pressures. You may use the vacuum also to do such things. Okay, so many targeted parts could be happens. As we know, yes, and yeah, bacteria. Those does not like this kind of oxygens. We're going to die, and we don't want to die them. We want to cultivate them. Yes. What could be the applications of such anaerobes? Can anyone say? Why we are interested in anaerobes? For degradations, but generally it's done in open house. What degradation? Hostels. Okay, so you are meaning that they are doing the anaerobes. Okay, fine. Nitrogen fixation. Okay, fine. Any else? Anaerobes, and maybe in a first year, first paper somewhere, application of microbiology. That points maybe you have learned into its aerobes and anaerobes may have came. Yeah. Yes. Sewage treatment. Okay. So some important process is fine. It is like a sewage treatment. Yes. So anaerobic digestions you are doing. Okay, fine. So where some particular process are there, some in the fermentation technology. Totally strictly the anaerobic condition is there. Okay, in a sewage treatment also anaerobic bacteria is a must. That is a some catalytic activity is generally done only by the aerobes. Some are with related anaerobes only. So that part should be get treated with the anaerobic condition. Also, like when you're looking, just I have told about the methanogens, right? Like a one gobar gas plant, right? Over there, we are working only with using the anaerobic microbes only because it's a totally closed system, right? There we are, that is a natural gas formation is done only by the methanogens. If you want to cultivate the methanogens, we need to maintain such condition, right? So in certain cases, so as per our applications or where we come to know that anaerobes are going to grow. Other than that sometimes we are going to learn about the basic concept related to preservations, but before it, like when we are looking to preserve our certain foods, jars are there, like a kitchen related, so many things are coming, like ketchups. It is totally closed in sterile conditions, but the same, it is an opportunity for the other microbes like anaerobes can grow now. You have closed it, right? Like sometimes you are having the some expiry date in every sort of the product. Okay, it is totally closed in sterile conditions. You have completely a wash bottle is been filled, but why they are coming is expiry date, or there is a chances of getting contaminations. Now the opportunity for the anaerobes may start to grow. You have maintained the total system related to the anaerobes. The oxygen will not grow. Now those oxygen haters are became favorable for it. So to study them, that is the like they are pathogenic maybe. To study them, we need to cultivate such bacteria inside our lab. So we need to create the anaerobic conditions for them first to start our experimental process, right? So you are telling about the anaerobe or the fermentations technology or the others also wastewater treatment and anything else. When you are looking to perform into lab scale first, you need to provide the aerobic conditions to anaerobic condition. Okay, so how you can acquire such anaerobic conditions? Only by getting certain technical advancement into that area. So first is the medium. Second, we are going to learn the some basic tools are being used, certain candle jars and others. So we can keep the certain capacity at that level. We can work it out into anaerobic condition. Okay, so such environment is just as it is the same pre-reduced media. That is some concept we are going to learn one by one. Three procedures are there. There is a pre-reduced media, anaerobic chambers, or anaerobic jars. So you going to work it out in the anaerobic chambers. Okay. There, what generally anaerobic chambers are there? It is like you are working in the laminar airflow, right? So in a laminar airflow, maybe it is a picture is there on the next step. Just let me show first one. Yeah. Anaerobic chambers, fully closed from the outer side. Here the places are there. You can insert your hands. Okay. So what is been done in general in this case? Everything is kept inside first. By using the vacuum pump, you are going to take out all the air from the outer side, and then you are going to feed either CO2 or the nitrogen. Okay. Nitrogen gas has been filled. The tanks are available. Okay, industrial scale nitrogens are available or the carbon dioxide are available. So you are going to feed that within it. So chamber is going to fill with the carbon dioxide only. So such jar is being used or the chamber is being used for the aerobics bacteria to be get cultivated. Okay, so what is generally done? It is the pumping part. Here the chamber is there. You can do your experimentations over it. So you can control very easily because here the filters are there. From the filter CO2 is coming. So if you are not going to keep the any specifically burner also, you can work it out because there is a no possibility of the other bacteria from the environment to get move inside. So such feasibilities also are there. So let's see one by one. I'm going to talk about that in detail also. Yeah. So pre-reduced media. So in the case of pre-reduced media, what we are going to do? We may boil for several times. Same medium or the liquid is boiled for several times. That is the best options. Or other than that, reducing it like a cysteines are being added further. So sometimes we know that it is like oxygen, it may yet remain. So it's going to reduce and convert it into water from O2 to chances of getting the H2O molecules. So reducing agents are being used. So freely available oxygen is going to get reduced into form of a water. So media is prepared in a way that the level of oxygen is going to get reduced as much as lower. Yes, in case of anaerobic conditions also, we may have the liquid culture and solid culture. But in the both the cases, we are going to keep them in static condition. We are not going to make them in a rotations more. Yeah, in case of liquid medium, sometimes rotation is preferable. The reason is what? That they are not going to stick at the bottom at least. It should get touch and utilize the whole medium. So may we going to give the agitations, not a aeration. Okay, here we are not going to use the cotton plug. We are going to use the cork. That is a total closing is done. Okay, or we may use the shaking flask or the other specific flasks are coming or the bottles we are going to use. So you can close it very firmly. So there is no chance of getting the water, air should be get transferred. So we may go for the aeration. Okay, sorry, agitations, not aeration. Agitations means you are going to shake it. Shaking may be there, but without addition of the oxygen. So you are going to limit it into this part. Yeah, other than oxygen-free nitrogen is bubbled through the medium to keep the anaerobic condition. When you are going to prepare it, what's going to happen that water having the capacity to dissolve the atmospheric gases. So while preparations, you may start to bubble the nitrogen gas within inside. So what's going to happen? The water having the certain level capacity to dissolve certain. So nitrogen is going to get dissolved into this area. To get the oxygen is going to get reduced. The first target, you have boiled it, so water is O2 is released. Scant amount of oxygens were there, you are going to use the reducing agents. So it converted into water. Now the third chance is there, the water may going to dissolve from the atmosphere again. So before it, you start to feed with the nitrogen gas. So nitrogen gas is going to get dissolved. It not permit or allow the oxygen get dissolved because the space is already been occupied. So you are going to use the dummy parts. Okay, dummy candidates, it's been used. before going to get oxygen get placed, we are going to introduce the dummy as a nitrogen to get their place. Okay, so these are the some basic steps into get media to be get prepared. Yes, other than that, media is transferred to the tubes with a which are being flushed with oxygen-free nitrogen, stopper tightly and sterilized autoclaving. During the incubation, these tubes are constantly flushed with oxygen-free carbon dioxide. Sometimes, yes, medium is prepared and transferred in the within the tube also, and flushed with the oxygen-free nitrogen or oxygen-free carbon dioxide. In a continuous rotation and mode, what's going to happen? It's going to become totally free of the oxygen. One step was the nitrogen has taken the place. Other than we are going to fed with the carbon dioxide. So there is a capacity of the nitrogen gas to get dissolved. It is not like a fully 100% is going to get occupied. So other capacity is yet remain for the water. So we are going to add O2 and CO2 almost having the same capacity to be get dissolved. So remaining part, that is the nitrogen, was not able to fill that full area. It's going to be now completely filled by the carbon dioxide. It is like our hemoglobin, O2 and CO2. It is continually transferred by our hemoglobin. Okay, in a RBC. So there is a capacity. Maybe you have learned the hemoglobin, that is oxygen curve is coming in your syllabus. No worry. That is part of the biochemistry. So it is related to this. So O2 and CO2 are having in case of our water level, it is about the same capacity there. So nitrogen is not able to fill that part. So what we are going to do? We are going to fed with the carbon dioxide. So all the space for the dissolve of the getting the oxygens to be get dissolved again, it's going to reduce. So we can use some medium. Okay, that's going to become the free of the oxygen. I hope this part is clear. How to get the oxygen-free medium? First step, boiling, oxygen is gone. Second step, scant amount of the oxygens may remain. You may going to add the reducing agent. So it's converted into water. You are going to feed with the nitrogen and carbon dioxide. So capacity of the oxygens or the capacity of the water to get the dissolved the atmospheric gas is going to get reduced down. So this is the final targeted part for the media. So media preparations, we are not going to work any case of anaerobic medium in our lab, because to maintain and work, it is very much costlier affairs. We don't have the such anaerobic chamber even. Yes, we can do certain level. I'm going to say about the candle jars and other basic experimentations can be done, but not right now. That is in master, because to handle the aerobes, it is for you maybe a difficult task right now. If I'm going to give the anaerobes, it is just a fun, no results. I just need to congratulate only. Okay, so I'm not looking to do it. Okay. Yes, the same part is related to reducing of the reducing agent. Okay, so what's going to happen? So reducing agents are being added in a way that they may change the color of our media. Whenever you're going to add the reducing agent, if the oxygen is present, okay, so it's going to reduce into water. So while the reductions, if it is a yellow color, it may convert into blue color, green color, or the brown color. When our media is going to have the brownish color more at the larger, you can confirm that now it is available for the anaerobic culture. Okay, as we know that while preparations, in the case of you have boiled it, you have added the some reducing agent, it must convert it into some different color. It is like a methylene blue. It start to get oxidized. Okay, oxidization methylene blue is going to reduce our oxygens to in a form of a water. So either it's going to become a colorless, or it may going to get the some different kind of a color. So we are going to add the reducing agent, which are in a form of a dye. Okay, reducing agent itself is a kind of a dye itself. It having their own color. When it get reduced, its color is changed. So you can confirm that my medium is now ready for conditions like anaerobic bacteria. So you cannot open it and check it whether its oxygen is there or not. But visualizations is done in a way that for the confirmation that you have added the reducing agent and it's converted oxygen into water, because scant amount of oxygen is going to remain if you're going to feed with oxygen, even with the carbon dioxide. That a scant amount of oxygens we need to remove. Okay, for the removal, we are going to use the reducing agent. Reducing agent having their own color, and that color is going to get changed. Okay, so this is the very indicator. So it is, we are going to use the indicator dye. It is called the indicator dye to confirm that now it is became oxygen free. Okay, it's became the oxygen free and easily can be used for our experimental purpose. Understood? So why we are using the indicator dye? The reason is to confirm about the medium is ready for the aerobic to now anaerobic culture. Fine. And during that time also, we may going to add or the flush with the carbon dioxide and nitrogen. Okay, so dissolved oxygen is has to react, because when the bubbling is going to happen, there is a chances you are doing certain experimentations in chemistry also, right? So within the chemistry, you are going to have sometimes of plastic, the tubes are there, you are going to mix. Tubes also been given, you may rotate or the invert it. The same principle is going to be held here. It is the combination of a chemistry with the microbiology. Is why it is required. Specifically, one of the example is related to methanogens. They are going to get die or kill just a scant amount of oxygen is there. So to grow them, methanogens are generally participating into what? For the biogas productions. Okay, for the biogas production, methanogens are major and they are highly sensitive with the oxygen. So we need to give the anaerobic conditions to them. So we need to manipulate our media in a way that it's going to become oxygen free. So how we can do it? One is the candle jar. Here you can see that you have prepared the medium. Okay, you have done into your work. You can maybe sometimes we directly don't have a facility like aerobic to anaerobic chambers. So what you can do, you can work it out into laminar airflow. You may have the negative pressures to as much as reduce down the oxygens from the outer area, but it is limited for you. So for a while, if you are going to transfer the some cultures and loops nearby the burners, okay, and you are looking to transfer, so those who are not that much sensitive, other than the methanogens, they can tolerate oxygen for a certain time period. Okay, so what you can do in this case, you may work it out. You may just run the sticking or something, but while keeping them in anaerobic conditions, we need to make it oxygen free. So what is done here? You can see the some tubes are there, plates and everything, right? So you have done the some cultures. Here you can see the tubes are with a cork. Tapering કરીને એક round વાળો છે, બરોબર છે. It's having the some are having the some their caps. So cap થી એને આપણે lock કરેલો છે. So you have performed your experimentations for a while within outside. Okay, in a chamber you have done, and then after you are going to either may have done in the anaerobic conditions or anaerobic chamber itself. But when you're going to take it out from that chamber and going to keep outside, there is a chances that it may come in contact with the air. So what we need to do, we may take the larger jars. What kind of a jars are there? It is like a you are using for your keeping your jaggeries and others. It is a glass jars are there. It may be a round or the square. Both kind of a jars are available. So what you are going to do, you are going to keep everything within inside, keep a candle within it, burn it, and then close the lid. So what's going to happen within the fraction of time, if the oxygen is there within inside, within the jar, there is a chances that oxygen will be there. It is going to get used by our candle. We know that candle is for the burning of the fire, it needs the very particularly the oxygen. For a certain time period, oxygen is going to get exhaust. As the oxygen gets exhaust, our candle is going to get disintegrated. Okay, their fire will be get disintegrated, exhaust. So what's going to happen? Oxygen is totally used up. Now our jar is free of the oxygen, free from the oxygen and now our bacteria are going to remain in anaerobic condition. Understood the things? How we are going to prepare the jar? Keep everything, whatever you want to keep your, that is a plate, tubes, or everything is there within the jar. Just keep the one candle, just light up and close their lid. Within shortest time period, it's going to utilize the whole oxygen. Okay, and their candle is the burning stage is there going to wipe off. And then you can keep as this tube in an incubator because you want to keep as a 37 degrees centigrade. The same thing is there into one small documentary. Let me show about it. It is how much? 1 or 2 minutes only.
Fancy candle jar.
Yeah, you can see that she's doing work in open place.
Special packs
right.
Where you break a little ampule, stick it in there, and it ties up all the oxygen.
This is explaining that how I have prepared
but tried and true way of having basically a pickle jar with a candle in it, and we use the premise that when we seal this up, the fire is going to burn only as long as there's enough adequate oxygen in there. Then it will go out, and when it goes out, it has lowered the oxygen level inside the candle jar, and this is how we will incubate both the plates, once again, incubated upside down, just like you would in the normal incubator, and we also have the test tube. All these have been inoculated, hoping to grow strep. So we'll incubate them at 37 degrees C for 24 to 48 hours
so inside explaining that it will going to get so within shortest time period.
The candle will eventually go out when the oxygen is depleted. You could fill this jar all the way up to the top with plates. We have done that in the past when we've had a full class. Obviously, with this just being a demo lab, I only have one plate of each kind, the blood plate and the Mitis salivarius plate, and then the tube is the Todd Hewitt broth. And there it goes, it's now out. So now this jar is ready to incubate. So
oxygen is totally get used up at the within 1 minute. Maximum is a minute is required to get exhaust that oxygen. Right. So same this point of questions are related to the stick plates and power plate can be asked in the 6 to 8 marks. Same can be asked in the 4 to 6 marks related to candle jar or the anaerobic chamber. So you need to draw properly. Right. This kind of a jar, what I have shown, you need to draw, label it, and you need to explain it. Yeah, the explanation is very short, right? But you need to explain by yourself what are the needs of such things. According to the marks, you need to write up. You don't say that I have just gone through your slide and 6 points were written, 5 points were written. I have written over there. Okay. According to marks, you need to have the sum write up in a proper format. Okay. Huh? Unit 1 and 2, yes. Yes, 2.2. It's our 2.2 is running. Yes. हाँ थोड़ो portions જે બાકી છે ને જે ઈ નહીં હોય ઈ તમે કીધું તમે. I will show you in next class. So till this is clear related to candle jar. About 5 minutes left, so I'm not going to start for the next topic. Candle jar is clear. How to use this candle jar? Okay, then anaerobics and others are there. I will take you further into it later. So just I'm ending here. You can put your attendance into this link via this link.